Hola, buenas tardes. Vamos a ir empezando, si quieren pasar a ocupar sus lugares. Y los que están llegando, si quieren ubicarse acá más cerca, nos solicitó el doctor E Eiden que si se pueden sentar un poquito más al frente para poder ver las caras jóvenes de los estudiantes. No, los jóvenes que están allá en las últimas filas. Bueno, también son bienvenidos a acercarse. Todos son bienvenidos, pero especialmente a los jóvenes que están allá atrás. Vamos a empezar entonces, allá arriba en la cabina. ¿Sí? ¿Ya? Ok. Buenas tardes a todos y a todas. Es un placer para mí presentar al doctor E Eiden. So today it's my pleasure to introduce Dr. E Eiden, who is the chief of the section on molecular neuroscience in the National Institute of Mental Health Intramural Research Program of the National Institute of Health. He received his bachelor degree in chemistry from Northwestern University and his PhD in pharmacology from the University of Kansas. He did later on postdoctoral training at the NIMH Intramural Research Program with Michael Brownstein before joining the faculty in 1985, where he's currently chief of the section on molecular neuroscience and acting chief of laboratory of cellular and molecular regulation. His work at NIH is focused on stimulus secretion synthesis, coupling, characterization of the vesicular and secretory proteins mediating chemically coded neurotransmission, and signaling pathways mediating neuropeptide-mediated stress responses. Dr. Eiden has studied peptides and neuropeptide signaling in the central nervous system. He has studied peptide physiology from different perspectives, such as evolution of peptides, pharmacology, pathology in several diseases, or models, experimental models of diseases, in neuroprotection, stress response, drug addiction, among others. He has more than 300 publications, and he also has served a lot as a past president of the International Regulatory Peptide Society and the Catecholamine Society. And also he has done a lot of editorial work. He's been in the editorial work of several journals, such as Frontiers in Neuroendocrine Sciences, and as a senior editor in the Journal of Neuroendocrinology, and has served on the editorial boards of Journal of Biochemical Chemistry, Journal of Neurochemistry, Peptides, Journal of Molecular Neuroscience, and Science Signaling. So I'm going to stop here because he has many other distinctions and merits in his careers, so that it will take long to speak about him, and we are here to listen to him, not to me. So I am very glad that finally Dr. Eiden is visiting our Institute of Neurobiology, and thank you very much, Lee, for this visit. I hope you also enjoy this visit as we have. Thank you.
Thanks very much. Dr. Morales, thank you very much. Dr. Diaz, thank you very much for your hosting me on this day. I've enjoyed it very much. I'm taking the liberty, I've been given permission, I should say, to sit as I'm just recovering from influenza. And better if I fall, that I don't fall so far from high to low. Okay. So what I want to talk to you about today is how classical neurotransmitters and neuropeptides collaborate at the synapse for physiological threat responses. And as this is a neurobiology institute, as neurobiologists, you're really used to thinking about organisms from the top down, from the brain to the periphery. The brain decides and the body acts. And I just want to provide a slightly different perspective, if I may. And this perspective is that while neurotransmitters like GABA and glutamate and acetylcholine are extremely important, it's also true that peptides have been part of the neurotransmitter armamentarium for more than 500 million years. Only half of that time have there been chordates like us on the planet. And so from our standpoint, organisms are really feeding tubes that run around the planet looking for food, and then they look for other feeding tubes so they can mate and make more feeding tubes. So it's all about the gut. It's not all about the brain. Of course, the brain is incredibly important for the navigation of the feeding tube. So a little bit different perspective maybe than you're used to. So when we talk about the gut hormones, these were discovered in the first 3 decades of the 20th century. Secretin was the first to be discovered, and in a very simple experiment, the duodenum was separated from the rest of a dog except through the bloodstream. And then food was introduced into the stomach, and pancreatic secretion was measured. And it happens that the gut secretes something that travels in the bloodstream to control pancreatic juice elaboration in order to digest food. So these gut hormones are the key informants to the rest of the body as to how to efficiently prepare for fuel intake. Now, I'm not going to ignore cognition in the brain. I'm as fascinated by it as you are. But I just want to build up from the ground level, from the gut, the need for decision-making in the central nervous system, and how neuropeptides found in the gut are also found in the brain and also help to direct the circuits that govern behavior. I like to think of the decade of from 2010 to 2020 as the decade of light. We had so many advances in microscopy, in fluorescence, and now we're entering the circuit revolution, where we can use genetic tools and light to understand how circuits in the brain work. Just a few words about what neuropeptides are. Some days ago, I gave a talk that was primarily on PACAP, and I got all the way to the end of the talk, and somebody raised their hand and said, "What does PACAP stand for?" I said, "Oh, I guess I made a mistake. PACAP is pituitary adenylate cyclase activating polypeptide." And the reason I don't emphasize that long name for PACAP is because although PACAP was discovered in the hypothalamus as a peptide that's released into the portal circulation to affect pituitary hormone secretion, almost everything I'm going to tell you about has to do with PACAP's role as a neurotransmitter, not as a hormone or a hypophysiotropic hormone. And that's because PACAP started out life as a pro-hormone that contained both PACAP, and it also contained another peptide, a growth hormone releasing factor. And together, these two gene products were involved in the release of growth hormone and the regulation of growth hormone in a series of vertebrates and prevertebrates. By the time we get to mammals, PACAP has only one other peptide associated with it called PACAP-related peptide, and we can't find a receptor for it. It looks like the PACAP-related peptide has just evolved itself out of a job in mammals. And that's another important point that I want to make. I know that you have, I'm very fascinated, Dr. Diaz, about the way this institute is set up with different realms of influence, including developmental. And in order to understand neuropeptides, you really have to understand not only evolution, but also the development of organisms. But in mice and rats and humans, it looks like PACAP is the only important hormone in this pro-hormone precursor. Now, why do I take the trouble to show you the pro-hormone precursor? For this very important reason. At low frequencies, and there's some debate about this, and you may study an animal species in which neuropeptides are released from large dense cord vesicles at low frequencies. But in general, the small synaptic vesicles that have acetylcholine, GABA, and glutamate are released from low to high frequencies of stimulation, whereas the neuropeptides found in the large dense cord vesicles are released preferentially at high frequencies. And this is why Thomas Hirschfeld calls neuropeptides transmitters that speak the language of urgency, emergency. Neuropeptides are released when an animal has to be able to act under adverse circumstances. But I mentioned the large dense cord vesicles because it's in those large dense cord vesicles that PACAP and other neuropeptides are processed from the pro-hormone down to the mature peptide. And what that means is, unlike the glutamatergic and the GABAergic and the cholinergic vesicles, which cycle at the nerve terminal, they empty and then they fill again at the nerve terminal. Empty, fill again, empty, fill again. Glutamate needs some help from astrocytes in the neighborhood, but in general, this is the domain where the classical neurotransmitters work. Neuropeptides, once they're released from large dense cord vesicles, in order to get more neuropeptide, it has to come all the way down the axon from the cell body, newly synthesized peptide. And we keep that in mind. We keep that very firmly in mind when we try to think about what is the neurotransmitter job of glutamate, GABA, and acetylcholine, and what is the neurotransmitter job of neuropeptides. Neuropeptides have to act in longer time frames than the classical neurotransmitters. So keep that in mind. This is nothing more than a dendrogram of all of the G protein coupled receptors. And I use this slide just to remind myself and remind you of two things. Some 40% of all therapeutic agents, what our Food and Drug Administration calls drugs, substances that are given to people for therapeutic effect, 35% of those act at G protein coupled receptors. And more, we're finding drugs for diabetes, for obesity, that act at neuropeptide receptors. In particular, I'll just point out a couple of those. There's my favorite, PACAP, up there, and it's part of a family called family B. Secretin, the very first hormone, is also in that family, as are GLP, a hormone that releases insulin and is very important in diabetes. And there are some highly successful new drugs that are based on action at the GLP-1 receptor. So these are all the peptide, these are just a bunch of peptides, and what they all have in common is those are their receptors. But the receptors for neuropeptides didn't all evolve in one clump of that tree, as you noticed. They were scattered all over the place. And this has been the evolution of G protein coupled receptors. They didn't evolve according to their ligands. They evolved according to the different tricks that they can do. And what are those signaling tricks? Those tricks are that when the peptide binds to the G protein coupled receptor, it can engage a G protein like GI that inhibits cyclic AMP formation, a G protein like G alpha S that increases cyclic AMP, and a G protein like GQ, which increases diacylglycerol and increases intracellular calcium. So these GPCRs and the peptides that activate them are metabotropic, as opposed to the ionotropic receptors for glutamate, GABA, and acetylcholine that bind directly to ion channels and immediately cause sodium or calcium to enter the cell in micro to milliseconds. The neuropeptides act in seconds to minutes, sometimes even over periods of hours. So that's an important concept to keep in mind. Now, another concept I'm going to introduce now is the concept of homeostasis and allostasis. And I don't want to get fancy with the definitions of words. I think you all know what homeostasis is. It is an animal maintaining its internal milieu so that pH is not extreme, so that blood oxygen content is normal, blood carbon dioxide content is normal. But there's a higher form of regulation called allostasis. It takes a brain. Homeostasis can occur in an animal that is unconscious, but allostasis requires a brain and consciousness. If an animal, the example I like to use is that if a zebra is thirsty and it goes to an oasis and there's a lion at the oasis, the zebra will not run out and drink. So its homeostatic mechanisms tell it, go and drink right now. And its allostatic mechanisms say, you better wait until that lion goes somewhere else. So that's just a homely example of the difference between allostasis and homeostasis. And you'll see in a moment why this is an important distinction. Now, forgive the fact that this is kind of a complicated scheme, but you're all familiar with the basic physiological underpinnings of stress. I told you about homeostasis and allostasis. And now I'm telling you that there are stressors that are psychogenic, that require consciousness and are involved in allostatic responses. And there are systemic stressors like hypoglycemia, inflammation, cold, and heat that don't require consciousness and that mediate homeostatic responses. Now, we think of the adrenal gland as the main output for stress, at least for endocrine stress. Okay, endocrine stress. We're also going to talk about behavioral stress and behavioral outputs. But for endocrine stress, if the animal is stressed, somehow that signal reaches the brain and the splanchnic nerve fires and catecholamines are released from the adrenal medulla, the fight or flight reflex. And at the same time, the hypophysiotropic adrenal axis is activated through CRH releasing ACTH and going to the adrenal cortex and increasing the synthesis and secretion of corticosterone in rats, cortisol in humans, So we just call it court. Now here's the part where I beg any of you who are students, you will see a picture of the autonomic nervous system, and you will know that acetylcholine is the preganglionic transmitter, and then norepinephrine is the transmitter in the sympathetic nervous system, and epinephrine is the transmitter or hormone released from the adrenal medulla. But there are other neurotransmitters, bona fide neurotransmitters, and PACAP is one of them. PACAP is found in preganglionic neurons. It's found in the same nerve terminals as the vesicular acetylcholine transporter. So if I were to blow this up a lot more, you'd see that acetylcholine and PACAP are in the same terminals, but PACAP is in the big vesicles, the LDCVs, and acetylcholine is in the little vesicles, the SSVs. If we stress an animal by inducing hypoglycemic shock with insulin, epinephrine levels go way up in a wild-type mouse and eventually come back down, much less in a PACAP knockout mouse, because PACAP is important in this pathway. And indeed, the PACAP knockout mice do not survive the hypoglycemia unless we actually inject PACAP into them. So PACAP released from this nerve terminal, and I'm not going to show you all the evidence. Those of you who are skeptical are going to say, hey, you knocked out PACAP from the whole animal. How do you know that just this PACAP released here is acting for epinephrine secretion? And we know that because our colleague, Corey Smith, has taken out the adrenal gland, where all of the PACAP in that gland is in the preganglionic nerve terminals. And if he compares a wild-type gland to a knockout gland, what he sees is that the wild-type gland will secrete if this nerve terminal is stimulated. This is just an adrenal gland, a slice of the adrenal gland, sitting all by itself in a dish. And if we add PACAP, we release catecholamines. If we add acetylcholine, we release catecholamines, but that quickly desensitizes. And the most catecholamine release at high frequencies of stimulation that are associated with stress are due to the PACAP, not due to the acetylcholine. So PACAP is a bona fide neurotransmitter at this synapse. Now let's go forward into the brain. And now we're going to look at the HPA axis. And we notice right away that stimulation with LPS doesn't require PACAP, but stimulation by restraining the animal does require PACAP. And if we do something called a social defeat test, where this animal is an intruder, the animal with the black coat is an intruder, put into this cage, then we lift this up, and this animal defeats this animal. Shows him in no uncertain terms he doesn't belong in that cage. And court levels skyrocket on the first day. And they keep going up with the wild-type animals for 14 days of social defeat. But the PACAP knockout mice, after that initial surprise, stress, these animals don't feel the stress anymore. And indeed, I'm just going to focus on one thing here, and that is there's an output that we think reflects depressive behavior, and that is whether animals interact with other mice interact with other mice. So in this little cage, there's a mouse, and this little cage is empty. And these are wild-type mice, and you can see they spend as much more time around the other mouse. They're interested in it. But after stress, they're not interested. They just keep to themselves. They're very anxious. Whereas the PACAP knockout mouse behaves the same, whether it's been subjected to just home cage conditions or social defeat. So this really got us interested in possibly integrating neuropeptides into a more comprehensive regulatory picture with classical small molecules. we saw that PACAP is released along with acetylcholine. In the splanchnic nerve, where is PACAP being released in the brain, and what is it being released with? So I'm just going to go through this very quickly, just to tell you that we can use the vesicular glutamate transporter as a marker for glutamate expression. And we find that PACAP is co-expressed with glutamate in retinohypothalamic neurons. And what's very interesting about this is that, and I'm going to go to this just shows that PACAP is actually released from this synapse. And this shows something very interesting. Here's a wild-type mouse, but it's not a mouse anymore because we've taken out, our collaborator, Martha Gillette, has taken out the hypothalamus. Now the hypothalamus doesn't have any legs, so it can't run faster at night and less during the day and show a diurnal rhythm. But those neurons still fire in a diurnal fashion. And if we look at that firing rate, and if we give glutamate in the early night, this phase advances. And if we give glutamate at the late night, this phase is, this phase is advanced. It happens sooner, so we call it phase advance, and this is phase delay. And PACAP itself does nothing. But now look at this situation. These are PACAP knockout mice. You still get the glutamate phase delay. If glutamate, this is like shining light onto the retina, is given in early night. But if it's given in late night, nothing happens unless you add PACAP along with the glutamate. So here's an example of a classical neurotransmitter working right along with a neuropeptide to achieve a regulatory control that can't be achieved without both of them. This is just a sagittal view of a mouse brain that is stained for PACAP and V-GLUT, or PACAP and V-GAT. These are markers for inhibitory neurons and excitatory neurons. I told you in the splanchnic nerve, PACAP is co-released with acetylcholine. In the retinohypothalamic tract, PACAP is released with glutamate. What about all the other places in the brain where there's PACAP? And what Professor Limei Zhang and her group, who are close collaborators with ours, determined is that, in fact, in the main business part of the brain, not so much in rostral, where PACAP is found in inhibitory neurons, or caudal, where PACAP is found in inhibitory neurons in cerebellum, but most all of the PACAP throughout the rest of the brain is co-expressed with a vesicular glutamate transporter, and therefore PACAP is a co-transmitter with the excitatory neurotransmitter glutamate. So now we're going to focus on some stress neuronal cell groups. And you'll forgive me, I'm going to say in some detail about the genetic techniques we use. Most of you are very familiar with these, and you may be bored by my explanation. But I just like to include it for those that aren't so familiar with these technologies, just so you can follow along the whole story. So first of all, we saw dissociation between the PACAP dependence of cord elevation and the PACAP dependence of hypophagia, animals eating less because they're stressed. We measure that as changes in body weight, but you can also just measure the food that they eat. They eat less food. And when we give a modest stress, like 1 hour, or a greater stress like 2 hours, or an even greater stress like 3 hours, we develop a PACAP dependence for cord elevation. But with respect to eating, these animals develop right away a PACAP dependence. Just 1 hour of restraint for 1 day is enough to make the animals not eat and lose weight, and that is blocked if they have no PACAP. What this tells us is that the PACAP that's involved in these endocrine effects and the PACAP that's involved in these behavioral effects have to be coming from two different places, or they would have a similar temporal profile. So we started looking for the different places where PACAP could be expressed, and we got some real surprises. PACAP, the highest levels of PACAP are, in most species, are found in the hypothalamus. CORT is released when CRH is released onto the pituitary, and ACTH goes to the adrenal cortex. So it would make sense that PACAP's endocrine effects would be mediated by neurons that originate in the hypothalamus. But that's actually not the case. When we look at a marker of activation after restraint stress, and the immediate early gene, FOS, is generally used as such a marker. I'm sure many of you are familiar with the use of FOS as a marker for neuronal activation. What we see is a PACAP dependence on the increase in FOS in the paraventricular nucleus, in the hypothalamus, and an increase in CRH messenger RNA that allows PACAP to keep this system going. But now what we're going to do is we're going to knock out PACAP in some highly specific regions. And we're going to do that with a Crelox tissue-specific knockout. And this is actually, it's a technique that everyone in neuroscience is grateful to have. And it's actually very simple. You can edit the genome of a mouse so that you put dotted lines on a gene that you're interested in our case, the PACAP gene. That gene will continue to behave perfectly normally until you express CRE in certain cells. And then the CRE will act as a molecular scissors and will cut out the functional part of the PACAP gene. And the reason this is such a powerful technique is that we can point to a region of the brain, and we can either use a virus or a particular type of genetic mouse, and we can knock out PACAP here, there, everywhere. And we see what the effect is on endocrine stress, and we see what the effect is on the behavioral effects of stress. And this, again, a lot of details, but the important thing is just that this is a PACAP knockout. But this is a PACAP knockout where we've only knocked out PACAP in the excitatory neurons of the forebrain, not in the hypothalamus, but in excitatory neurons that presumably project to the forebrain. And there's no effect on behavior, there's no effect on weight loss, but there's a profound effect on the endocrine axis, the stress endocrine axis. Okay, so I'm just going to tell you in words that we're really interested in this pathway because by doing retrograde tracing and anterograde tracing, Sunny Zhang has shown that this is a PACAPergic pathway that begins in the prefrontal cortex and extends down to the paraventricular nucleus to control endocrine stress responses, but no effect on behavioral stress responses. To look at the behavioral effects, we looked at a very specific nucleus in the brainstem called the parabrachial nucleus. And a number of laboratories have studied the role of the parabrachial nucleus, which is a nucleus that expresses the peptide CGRP, calcitonin-related peptide. And this projection goes to the extended amygdala, the bed nucleus of the stria terminalis, and one that might be more familiar to you, the central amygdala. And so we looked at PACAP in this system, and it is there, and that's been confirmed by a number of groups. And now, if we put a CRE-driven GFP and inject it into either PACAP flocks or wild-type mice, the CRE will, this is a promoter, this is a synaptophysin promoter, so it just means we'll be expressing GFP in all the cells. However, with the CRE here, will only knock out PACAP in the cells that we inject. And what we find is that PACAP projects to the BNST, it projects to the BLA very prominently. And if we knock out PACAP just in this cell group, the PACAP in the BLA and in the BNST disappears. So this is a PACAPergic projection, like the one from the frontal cortex to the hypothalamus, only this one goes from the parabrachial nucleus to the extended amygdala. And now we have something, I think, very interesting, which is that we have cells in the central amygdala that get inputs from PACAPergic neurons, and they somehow cause a behavioral change with stress. So what we have here is a kind of switch buried deep in the brain. What happens if we try to manipulate that switch to the behavior we're interested in? Does PACAP even matter, or does it not matter? Well, the PACAPergic neurons from the PBN project to cells in the extended amygdala that are called PKC delta cells. Doesn't matter, it's just a marker. Of course, we're very interested in it, but you can just think of it as a marker. It's a color paint that paints those cells, PKC delta. And what we find is that the PACAPergic projections go to those cells in both the BNST and the central amygdala, and don't go to other cell groups in that nuclei. So we're getting closer to a node, a synapse that forms a node that we can study and that makes a difference for behavior. And indeed, FOS activation in the extended amygdala occurs predominantly in these PKC delta neurons. So the FOS increase is PACAP dependent. The PACAP is projecting to these PKC delta neurons, and they're the ones where the FOS is going up when we restrain the animal for 2 hours. And the activation does require the expression of PACAP in the PBN. And here, these are experiments where I'm not going to go through all the different types of manipulations we did, but in this case, we can use a PACAP Cree mouse to express just in those PACAPergic neurons in a Cree-dependent manner, or we can knock out PACAP if we inject into PACAP floxed mice. So we have 2 ways to go with this analysis. And what we learn is that FOS activation and CRH, I'm going back to the hypothalamus, and I told you that that requires a cortical projection. This PBN projection doesn't do anything in the hypothalamus, completely independent of the endocrine stress axis. So now we use a technique that's called designer restricted expression exclusively to designer GPCRs. And these GPCRs are built around the GQ-coupled muscarinic receptor, or the GI-coupled muscarinic receptor type 4. So if you see M3, this is going to lead to activation of these neurons when the animals are treated with the designer drug. And if we're expressing this one, the M4 variant, then when the animals are treated with CNO, we're going to shut down those neurons. They won't be activated. And what we learned from these experiments by putting the M4 DREAD molecule in a Cree-dependent way into various types of mice, we can use PKC delta Cree mice. This is what Sunny chose to do. Luckily, somebody had made a mouse where Cree is expressed only in PKC delta neurons. So if she puts a Cree-dependent DREAD into the central amygdala, it's going to express this particular receptor only in the PKC delta cells. And when one does that, one finds that in the BNST, nothing happens. There's weight loss. But if we shut these neurons down, we don't get the weight loss with stress. It's significantly attenuated. And more recently, Diego Castillo Roland, who came to our lab from the veterinary medicine program at UNAM in Mexico City. He's been looking by optogenetic stimulation. In this case, what we do is we inject into these PKAPergic neurons, not just a marker that says you injected them, so you can look at the nerve terminals. What we're injecting are light-activated channels that when we take out slices from these mice and shine light on these terminals, and these are light events, we're shining light in very short millisecond durations, and we're getting a current flow that corresponds to GABA-induced currents because they're completely shut off if we treat with the AMPA receptor antagonist CNQX. Now what this means is that these neurons that contain PKAP, we know they need to release the PKAP to make FOS go up. We know they need to release the PKAP for the animals to show an anxiety-related hypophagia. You gotta have PKAP at that synapse. But if we just look at the synapse optogenetically, it looks like a typical glutamatergic synapse. So what's our next step? And I'm leaving you here just to, because that's where we're at. What we want to know is if we put a PKAP antagonist here, do we change this response, meaning that PKAP contributed to the glutamatergic response? Or is there another PKAPergic signal underneath that glutamatergic signal that is responsible for the FOS induction, that's ultimately responsible for these neurons being required for stress-induced hypophagia? So just to summarize, I told you about PKAP and acetylcholine co-release at the splagnico-adrenomedullary synapse. I hope those of you that are studying the autonomic nervous system for the first time remember that PKAP is indeed a neurotransmitter in the peripheral nervous system. PKAP and glutamate are co-released at the retinohypothalamic synapse, and PKAP collaborates with glutamate-induced phase advance in the suprachiasmatic nucleus. PKAP and glutamate are co-released at the parabrachial extended amygdalar synapses, and PKAP permits the behavioral response to restraint stress, as well as FOS activation. And finally, PKAP and glutamate are released at frontocortical hypothalamic synapses that mediate an endocrine response to stress and participate in FOS regulation and CRH biosynthesis there. So I guess my take-home point to you, and I appreciate your patience, is that understanding classical neurotransmitter, neuropeptide co-transmission is really probably going to be required to fully understand circuit-based behavioral decision-making in the brain. So thanks for your attention. I'm happy to take any questions you might have.
Thank you, Dr. Eiden, for this interesting seminar. And if you have any question? No? Lee is going to do a question.
Hi, Lee. Thank you very much for the beautiful talk. I just want to ask a detailed experiment about the chromaffin cell that you said the PACAP can be the main neurotransmitter. So how you, or do you have any experimental evidence that how the PACAP by itself can make the, eventually the adrenaline release, how it could be the mechanism, molecular mechanism?
Thanks for that question. There's some interesting new data coming out from Arun Thrakkaman's lab. And what he's done is to elicit PACAP secretion from chromaffin cells, either chromaffin cells that are wild type, and he gets release. Calcium influx, not sodium dependent action potentials, but a calcium dependent secretion. And if he removes a particular enzyme, phospholipase C epsilon, PACAP no longer can carry out that secretory function. So it seems to be metabotropic. It seems to be cyclic AMP dependent, and it may rely on a RAP-GAF protein called EPAC.
Thank you. It's a second question. It's a little bit philosophical. No, I know your point is the neuropeptide should also be considered as neurotransmitter. So you just explained one example, which is through also metabotropic pathway. So classically, you know, in the neuroscience community, people call neurotransmitter to the small transmitters which directly open ion channels, and the neuromodulator, which is through the metabotropic receptor pathway. So what is really the benefit effect that we call two different things with the same name?
Well, it's interesting that in the peripheral nervous system, sorry about that, in the peripheral nervous system, norepinephrine at sympathetic neurons has always been called a neurotransmitter. And dopamine in the brain and at small intensely fluorescent cells in ganglia has always been called a neurotransmitter. Dopamine and norepinephrine are neurotransmitters. And yet, neither dopamine nor norepinephrine are ionotropic. They are exclusively metabotropic. And so I think that we're going to have an adjustment. I think historically, those who have been a long time in the field of neurobiology and grew up with gray type 1, gray type 2 synapses. How does the brain work? By inhibitory and excitatory neurotransmission, inhibitory release of GABA, opening of chloride channels, glutamate interaction with NMDA receptors, influx of calcium, ionotropic. But in fact, there are a whole class of glutamate and a whole class of GABA receptors that are metabotropic. So I think we're going to have to me, the best thing to do is kind of abandon the phrase neuromodulator for two reasons. It's rather rude to dopamine and norepinephrine to sometimes call them neurotransmitters and other times call them neuromodulators. And in addition, the term neuromodulation, and those of you that work with, for example, slow wave encephalography and with transmagnetic stimulation, that whole emerging area of neurotherapeutics is called neuromodulation. So I think what makes the most sense to me is give the word neuromodulation to the people studying transmagnetic stimulation and just use ionotropic and metabotropic neurotransmission to talk about whatever is released at or near synapses from small synaptic vesicles or large dense core vesicles. I admit, for some of us, that takes some getting used to, but at least we should be clear when we have a discussion what we mean by neurotransmitter. Yeah.
At the beginning, you showed these zoological different classes. I wonder how extended is this idea of having, this is always something that always happened from patterns and other groups of animals. I suppose that's the case, but have you looked into that?
Do you mind trying again? I didn't really catch
I
that.
was just wondering how evolutionary conserved is this idea of having this, because you showed that at the very beginning of your talk. So if you can comment on that.
Well, PACAP, the primary structure of PACAP 1 through 38 is remarkably well conserved from very primitive organisms up to primates. And presumably there's a selective pressure that the PACAP and its receptor do something useful. But PACAP has 3 different receptors: VPAC1, VPAC2, and PAC1. And of those 3 receptors, another peptide, VIP, interacts at the VPAC1 and VPAC2, but not at PAC1. To understand the logic of evolution I think it's a question too big for humans. In order to mimic evolutionary time, we would have to have this conversation one word at a time and exchange words every 5 years. You see my point? So I don't know what the answer is for that kind of selective pressure. And I also don't understand why, if PRP no longer has a receptor, why is it conserved? And it is actually quite high, not as highly as PACAP, but it is quite highly conserved. So these are, as far as I'm concerned, these are post-Darwinian mysteries.
Thank you.
I have a question about time points. So you said neurotransmission is like very fast, even fractions of seconds, and neurotransmission by neurotransmitters, classical neurotransmitters. But in the case of peptides, it takes seconds to minutes. So I guess that's one of the difficulties of study secretion of neurotransmitter and peptides in the central nervous system, because there are neurons that have 1 neurotransmitters and 3 or 4 or 5 or more neuropeptides that release. Can you comment on that? And also, I think also the influence of other peptides that are coming from the periphery, like GLP-1 or leptin or some other peptides that come from the gut, also make this more complex. And in that sense, neurotransmission by peptides has been called neuromodulatory instead of neuron transmission.
Yeah, well, I explained as best I could why I think that neuromodulation is not the right word for neuropeptide neurotransmission. But I understand that some people will use that term. In terms of the time scales, I believe that optogenetics is now going to give us the opportunity, along with chemogenetics, to define what a transmission event is. For example, classically, in the fight or flight reflex. An animal is frightened, epinephrine is secreted over a period of minutes. Okay. So when we, and this is something that Richard Palmer and his colleagues have tried to do. So with the experiments that I talked to you about today, imagine the following. Imagine that we have restrained an animal for 2 hours, and that resulted in a change in behavior over the next 24 hours. Now I think we can ask, how do we mimic the restraint? Do we do it by injecting CNO 10 times once every second for 10 seconds? Do we inject CNO once every 10 minutes for an hour, and so forth? These are not simple experiments, but I believe they are experiments that are going to have to be done to define what is a neurotransmission event. If it's a glutamatergic event, how many times does it have to happen before it triggers a behavior? That's an important question for all of us, whether we're studying glutamate in SSVs or neuropeptides in LDCVs.
Okay, if there are no more questions, thank you very much, Lee. Thank you.